Hello, my name's Steven Petrou. I'm from the Florey Institute at the University of Melbourne. And today I'm going to talk about loss of function gene mutations in SCN2A and start to open the dialogue around the implications and opportunities for therapeutic intervention in this space. So to get started, I'd like to just spend a few slides in discussing what SCN2A does. And we know that SCN2A makes a very important protein that's called an ion channel protein. And when it's converted from a gene into a protein, which is what genes do, it is called NAV1.2. So SCN2A is the gene name. NAV1.2 is the protein that's made by the gene SCN2A. And what does NAV1.2 do in the human brain? And we know from years and years of work that NAV1.2 triggers electrical activity in neurons. And if we look at this screen below, and I'll just bring up a pointer, if we can look at this image down to the right here, this is a stylized version of a neuron or a nerve cell in the brain. And there are sort of three major parts to a nerve. There's the incoming signal part, which is called the dendrite, and that's shown in this green cloud. There's the actual cell body part of the nerve, which is shown here in red. And then there's this pointy structure to the right that's called the axon. And generally, information is sensed by the green area, by the dendritic tree. That information is essentially decoded in the somatic part of the neuron, and a decision to fire an electrical response in following this incoming activity is made at a very specialized part of the neuron called the axon initial segment. And that's been blown up here in this little window here. And you can see within this very specialized part of the neuron that there are lots of sodium channels. The NAV1.2 or SCN2A gene product is shown circled. SCN8A or NAV1.6 is also shown within these regions. And they have a unique distribution in a neuron, depending on the type of neuron, depending on the stage of development. And these sodium channels in the specialized part of the neuron are really what determine whether an electrical signal is initiated. And in neurons and other cells of the body, particularly muscle cells, when this happens, you can produce an electrical signal that is called an action potential. You may have heard about these before. And that action potential is the way that a neuron can send a local signal a long way. And that's what the axon is there to do. It's there to transmit the signal to distant parts within the brain. And every day we experience this because it's action potentials that allow us to move our limbs and pick objects up. They're our nerves that are starting from signals in the brain are ending up in our muscles all over the body. Those signals are carried by action potentials. And you can see because NAV1.2 SCN2A gene product is so exquisitely positioned within a nerve to control this function, dysfunctions of this gene NAV1.2 are conversely very well positioned to have a negative impact on human health. And this diagram on the left just shows that there's a very, very strong interplay between sodium channels and potassium channels seen here on the right. The action potential is shown in the red, which is when we blow up and look at the nature of the electrical signaling, it's around about 1/10 of a volt, to put into the terms that we may understand the nature of the electrical signal in the body, about 1/10 of 1 volt. And you can see underlying that is a very choreographed activity of sodium channels and potassium channels. And as many of you may know, sodium and potassium channels are two of the major genes that are involved in a whole host of epileptic encephalopathies and syndromes. KCNQ2 being a particularly famous potassium channel for being involved in human disease, NAV1.2 in some sodium channel disorders, NAV1.1 in Dravet syndrome, NAV1.6 here in other developmental and epileptic encephalopathies. So a very important part of a neuron, full of these very important genes whose dysfunction causes many of the disorders that we are unfortunately all to know too much about. And why is that important? Well, in a single neuron, we can understand that an action potential arising here will travel down, and that these little blue blobs are actually little blobs of fat that cover the neuron's axon, and this allows the signals to travel unimpeded. It's almost like an insulating layer, and that way the signals can travel distantly. And this axon can be hundreds, fractions of a millimeter, or even millimeters long, or even way longer, like a meter long when it's a motor neuron. So that this fat becomes very important in allowing the signal to move from the soma here, where I've got the cursor, all the way to the end. Now, why do you want to do that? Well, normally a signal travels in the brain. This area is the output structure of the neuron, and that talks to other neurons. And you can see here, this neuron 1, if an action potential comes along here, it talks to neuron 2 through these specialized structures called synapses. This neuron looks at all that signaling that's occurring and decides itself, and that'll be happening from multiple neurons will be impinging upon this, not just this one. It'll decide what to do with that information and then fire its own action potential to talk to the subsequent neuron. And it's this ability for neurons to communicate that is critical for the sorts of activities that we all take for granted every day when we think and act and do things. And you can see that if you impair this process through changes in the properties of neurons and how they listen to signals and how they generate their own signals, you can see why diseases like epilepsy and autism and other things may emerge if you're interfering with this very critical process of how neurons talk to one another. It's as though we've changed the language, and we know when we're communicating in our normal language that we know well, how efficient that can be. And if we then go to a foreign country and someone talks another language, we know how confusing and difficult that can be to do our daily activities. And it's the same thing in the brain. Subtle changes to the language of individual neurons affects the function of the entire brain. So we've learned a little bit about how changes in excitability that are caused by SCN2A are critical for the function of the brain. And now we want to ask the question, what is SCN2A loss of function, and how does it happen? We all hear about these terms, loss of function, gain of function, etc. But in particular, we want to talk about loss of function, which is the topic of today. So how do mutations arise? And I've shown 3 major genetic mechanisms that can result in loss of function of SCN2A. And these are namely missense mutations, secondly, nonsense mutations, thirdly, macro deletions. And I'll touch on each individual one of these. As I said, there's a so-called central dogma of molecular biology, and that is the genes make messenger RNA that makes protein. And it's really, you can think about genes as the master plan, mRNA as the working copies of drawings to produce something like a building, and then the proteins being the actual building that you make. And it's this 3-step process: master plan, intermediate drawings, which are the mRNA, final product, which is the protein. And each of these converts a genetic signal into a protein signal. And we make, somewhere in the order of 20,000 proteins in total to be a human. And the particular way that these proteins are expressed in various cells and they come together makes us who we are. And this is the signal up the top. You can see here that three chemical DNA signals made of these nucleotides encode for a single amino acid. And there are a relatively small number of amino acids in the body that are encoded by different sequences within your DNA. And this is a very old, from the 50s and 60s when we started to understand how this occurred. And we know now this is fixed, and this genetic code is a very well conserved process in many organisms. So now, if you have a single change in one of these DNA molecules, the so-called nucleotides, of which there are 4, one change can have an effect on the instructions for the protein, and that introduces a single amino acid difference. And this is a wild type. The below could be the mutant protein, and that can severely affect the function of this protein, such that it doesn't work as well as it did before. And for SCN2A, you can change the function of the protein so it works stronger and then gain a function, but you can also change the function of the SCN2A so it doesn't work as well. And that's called loss of function. And both of them can arise from missense mutations. And it depends very much on the nature of the change. Now, nonsense mutations, on the other hand, what they do is, rather than saying, well, I want an arginine or a leucine or a glycine in this position, you can imagine this like a string of pearls, and you've got 20 different pearls to select from, and you need exactly the right sequence there to have the right protein. And there's another thing that happens. Eventually, you need to say, stop, I've made the protein. I don't want to add any more amino acids to this. I'm done. And that's called a stop signal. So there's particular signals in our genes that say stop making protein, and now it's ready to go. Now, if a mutation inadvertently introduces one of these stop signals, the protein is only partial length. So you don't make the full length of the protein. You make what we call a truncated version. And these proteins tend not to work as well, because it's like making a car and stopping before you've added the engine and the doors, at which point you don't have a full car anymore. You've got anything that somewhat looks like a car and doesn't function like a car. It's the same thing that happens in the protein world. And these tend to produce proteins that don't work. So nonsense mutation produce foreshortened proteins that don't do the original thing they wanted. More often than not, a nonsense mutation will lead to a loss of function protein. The third mechanism is that we can have wholesale changes to our DNA, our chromosomes, sorry. Here in these two cases, I was talking about a random event that causes a chemical change, and this happens all the time in everybody. And these mutations can, in an adult, give rise to a cancer cell. These mutations, when you're forming the sex cells, the eggs and sperm, can give rise to mutations that can be passed on to offspring. And they can occur at all times, anywhere in between the formation of a gamete or a sex cell all the way up to an adult. And these mutations can occur through chemicals. They can occur through random events in the reproduction of cells and the creation of DNA. And it can occur because of random energy from various sources of radiation from the sun, etc. So there's lots of reasons why this happens, and it's random and it's not controllable. Here, similar things can occur, but you can impact whole regions of our chromosomes. And if you recall, chromosomes are just full of these sorts of genes, all wound up in a particular structure, and there's 23 of these chromosomes. But things can happen such that chromosome can break and a whole deleted region can occur. This is the normal chromosome here, and you can see this light blue region was deleted. And this chromosome can be now smaller than it is, and in this case, multiple genes may be removed. And if SCN2A is included in that list, you can actually, I'm not saying SCN2A is on chromosome 4, it's just an example that I found, but if you remove a chunk of a chromosome, you remove the genes and you have none of that gene. And so that also causes the fact that you've completely removed that gene means that the protein is not going to be made, and you're going to be, you're going to have a similar situation as you would with a nonsense mutation or a missense. All of these can lead to reduced function. And what happens at the protein level? So I've done this stylized drawing here. Each of these cylinders can represent a sodium channel in a given neuron. A missense mutation, because these disorders are what we call autosomal dominant, means that we have one normal copy of the gene and we have one mutant copy that's got the variation in it, and it's this version in orange that doesn't work. So half of the copies of the proteins we make are dysfunctional, and so we've only got where we should have had 6, for instance, we only have 3 functional copies, and that's because of a point mutation caused by a missense mutation has stopped this orange version from working properly. As I mentioned, truncating versions, because of nonsense mutations, can produce partial length proteins. These half-length versions also don't function well, and you're only left with 3 working copies. Chromosomal deletions or other types of mutations can result overall in the production of less protein. So you don't even have a foreshortened version, you just simply don't have it. And fundamentally, though, in each of these cases, you're left with a reduced complement of sodium channels. This overall produces reduced activity in the neurons because of loss of function through mutation, loss of function through deletion, or partial deletion, loss of function through complete deletion. So I hope that gives you a bit of a feel for genetically the consequences on our proteins. And if we think a little more about what the protein does, well, here's another image of a single protein, how we do it as a cartoon. And what our sodium channels do is they allow sodium to move across the cell membrane. So sodium on the outside of our cells can move inside the cell and to trigger electrical activity. And I've shown here, so this is a cartoon. This other one is a advanced version of a cartoon that shows how the sodium channel threads across the membrane of a neuron. And when we have, excuse me, we have very good evidence that this is the way the sodium channel presents itself in the neuron, and then it folds up into this complex structure on the right. And these are images taken from a method called X-ray crystallography that helps us determine the structure of the actual protein. So this is the cartoon. This is a very simple representation of the protein, how we think it's disposed of within the cell membrane. And actually, in actual reality, it looks like this. But in each case, sodium goes from outside to inside. Here, we know the region I'm highlighting here is where that movement of sodium occurs in the so-called pore region, which is shown by this little hole in the cylinder here where the sodium goes through. And there's 4 of those such regions in a single sodium channel protein. And in this crystal structure, again, the sodium goes from outside to inside. And if you look at this other view where we've rotated this sodium channel, you can actually see a hole in the middle. And so there is a hole like shown in the cartoon, and that's the hole or the pore where sodium moves. And all these other areas you can see here shown with these arrows are where we've found different mutations that cause either gain of function or loss of function changes. So all these mutations in the areas outside of just the pore can have a big effect on the sodium channel itself. So a single change in this whole protein is enough to cause either gain of function or loss of function. So this is what happens genetically. These are the consequences on the protein, the NAV1.2 protein. And if you reduce the function of this gene, you can give rise to various disorders. And when we look at the activity of the sodium channels, these are some of the methods we apply. And let's just focus on the black signal, which is one of the wild type or normal copies of the gene. When we apply particular methods to study it, we'll see at the very fundamental level, the sodium channels, this is their functional signature shown here. And at the neuronal level, this is what they do. They enable the firing of action potentials. But if you look at this mutation, R853Q, which is an arginine to a glutamine at this position within the protein, these currents, the sodium channel is smaller. You can see here, the peak is less than here. This is a loss of function mutation. And when you look at the neurons, you don't get as much activity. So that's and this is what we see in this particular way of studying it. But let's just pause on this and more on this concept later. This is what happens at the most fundamental level. Not enough sodium channel, you don't get enough neuronal activity. Each of these upward deflections is one of those so-called action potentials, and you need to have a normal complement to have normal function. And you can see here, you can have far fewer if you've got a loss of function mutation. So what happens to people that are carrying these loss of function mutations? And this diagram really sums up the clinical spectrum of SCN2A disorders, and it can be broadly characterized into patients that have got gain of function and patients that have got loss of function. And today, where we're talking about loss of function, I'll focus our efforts here, but you can see you can get specific disorders because of gain of function. But if we look within the loss of function groups, we can see that there are two major phenotypes that we see in the patients. Autism and intellectual disability caused by mostly these nonsense or truncating loss of function mutations. And the other disorders, we see a late seizure onset developmental and epileptic encephalopathies. So here, as in contrast to the gain of function mutations, the seizures can occur later in life. And rather than occurring often on the first day of life, as you might see with gain of function disorders, here they occur after 3, 4, 5, 6 months, or even later. And a very different type of seizure may present. These patients and people may also have hypsarrhythmia, which is a movement disorder, and pharmacologically, often these seizures can worsen with blockers of sodium channel. And there is a split though. There's patients that seem to get more an autism phenotype, and there's patients that more get this developmental and epileptic encephalopathy phenotype. And maybe more on that later is why does a loss of function mutation lead to autism in some patients and this DEE in other patients, and a little bit more on that later. So, and that's a prelude to this. So why do SCN2A loss of function mutations cause disease? And there are three questions that help us in framing that particular question, that overall question. We've got this whole issue, gain of function versus loss of function. They do share some aspects of the disease. We can see in gain of function and loss of function kids, seizures in both, but not others. Movement disorders tend to be more prevalent and stereotyped in the loss of function kids than the gain of function kids. So there's something about gain of function and loss of function that results in aspects of the disease that look similar. They both have developmental delays. They both can present with seizures, but then there are other things that make them different. Why? I mentioned this before, loss of function developmental encephalopathy versus loss of function autism. Why do kids with protein truncating and certain missense loss of function mutations have autism and often without seizures? And why are they different to kids with certain types of missense loss of function mutations? That's a really good question that we don't fully understand yet, but a question that we need to keep asking. And then the third and a really important question is, well, if SCN2A is so important in causing electrical activity and excitability, why does the removal of an excitatory component eventually lead to epilepsy? It's like saying, well, I've cut the cable to the accelerator in the car, but it goes faster than it did before. And that's a great puzzle to the science. And there's a few clues starting to emerge that might explain that. And the studies, and this sort of summarizes that data. And what we're calling it is a paradoxical increase. So it's not we didn't anticipate that removal of an excitatory element would cause an increase in neuronal excitability. And this was a study that's recently been published by the group led by Kevin Bender. And what they did, they studied 3 groups of mice. In the black, mice that had a full complement of sodium channels, SCN2A channels, mice in blue that had only half a complement, and mice in red that had no SCN2A. And what they found in the animals that had no SCN2A, that excitability of the neurons, as shown by this upward shift of this red curve, was greater than the other 2 groups. And could this explain, and mice are different to people. People with a single deletion of SCN2A might experience the red shift. So we don't always see a full correspondence between mice and people, but sometimes we do. We've got to bear that in mind. But there's a clue that if you remove SCN2A, you get this paradoxical increase in activity. This other group by Dr. Yang also found this in their model, that they found that animals with both copies of the SCN2A gene removed had this paradoxical increase in activity of those neurons. And the question really is this what gives rise to the epilepsy in the patients that have this removed? And as I mentioned, though, loss of SCN2A is a very common genotype that we see in patients with autism, yet they don't always present with seizures, but autism patients do present with autism phenotypes. And one of the correlations that was also found in these mice that had the SCN2A gene removed is that another part of the neuron, that dendrite that I mentioned before, has different function. You can see here the black is the normal cell, and the blue and the red is the mice whose neurons had half a copy or zero SCN2A. And you can see here that function of the dendrite, and I won't go into the details of this experiment. It's not that important for overall fundamental understanding of the mechanisms here that we're talking about. But something's different about this neuron that looks like a loss of function. Here on the left, the axons function with gain of function, but the dendrites, the receiving part, functions with loss of function. Now, the question that arises, is this gain of function shown in panel B and panel C epilepsy? Is panel F autism? That's what these authors are putting forward as the concept, that this may be the way that the disease emerges and why we get the different aspects. Now, it's not 100% clear that this yet happens in people, and we always have to bear that in mind. These are animal models, but these are really, really important first steps as to how we think about how diseases emerge, and then it impacts how we think about treatment. Because now we've got this paradoxical increase in excitability. What happens if we try to correct the gene deficit, which is not enough SCN2A, and we start to tip in more? Would we make the epilepsy worse? We can fix the autism by increasing the amount of SCN2A. We can see the red and the blue, there's not enough to give us this black signal that we need. So we can tip in more SCN2A, maybe restore that, but maybe we overplay our hand and we make the epilepsy worse. So these are the things that we need to consider when we're looking at how we think about therapy. When does this happen in the lifetime of a person that's got one of these mutations? And if we treat early, can we avoid this from happening? If it's already happened, is there a way to treat that can slowly and progressively increase the SCN2A in a way that the body's natural systems start to restore this function and go back to the function of the black cell? You can see here, we can paradoxically reduce the activity. And how do we go about thinking about those therapies? And what are the options for these people? And and this is a really important slide. And as all of the things that I've told you, and at a very fundamental level, we might expect that people with SCN2A loss of function would simply require therapies that either enhance the activity or the expression of SCN2A. So make the remaining cells channels work harder, or add more channels to the mix. But then this question arises: but if there's increased excitability, as suggested by the mouse studies, how do we think about framing these therapeutic studies at a clinical trial level? And what I've done here is really just made a list of ways that we can think about increasing function. Within this list, there are activities that are happening across the globe in laboratories and in various companies that are interested in trying to tackle loss of function mutations in SCN2A. And if you look at these lists, what we can develop drugs that directly support the function of SCN2A. So this is a form of a drug that makes the existing SCN2A or NAV 1.2 channels work harder. And there's been work that's shown for Dravet syndrome, which, like autism, is caused by reduced function of NAV 1.1 or SCN1A. Studies have shown that if you can enhance the function of the protein, you can have an impact on the disease. You can say drugs that don't directly impact the SCN2A channels, but indirectly support the function of the neurons that are struggling because they don't have enough SCN2A. And you can support that via different mechanisms by supporting various functions at the neuronal or network level. You can think about drugs that increase the amount of that signal molecule, the plans, mRNA, so that you end up making more SCN2A ion channels. You can think about drugs that say, okay, we only have a fixed amount of messenger RNA because the other copy has got a mutation, but can we just direct the system to make more NAV 1.2 channels from the plans that we've got? So we've got one set of blueprints, but we're going to make multiple buildings from that one set of blueprints. And so we can have the same number of buildings as we otherwise would have had, even though the other copy of the blueprints had a photocopying error in them, for instance. Maybe we can use a gene therapy that simply inserts more copies of SCN2A into the brain and into the right cells of the brain. And that's another way you can think about it. Let's just add them back through a gene therapy. Use a packet into a virus, inject it, and then overcome all the issues that you need to for such a therapy. But if you can overcome those, you can increase the expression. We've all heard about gene editing, and there are various approaches to that. CRISPR is one of the ones that's quite famous in that area, but there are other approaches that you can do where you simply fix the mutation in the brain, and then you let nature take its course. So let's go in, repair the SCN2A genetic mutation, and then the body will then naturally start to produce the right amounts of the right protein to correct it. And the other way is, can we use cells that are complementing the function in the brain to overcome the balance of excitability and inhibition to support. And there have been studies that have shown that this can be effective in epilepsy as well. So these are just some of the approaches that can be trialed to come up with a fundamental concept to deliver therapy. As I said, many of these have been explored by labs and commercial entities around the world, which gives us hope that something will emerge. And as you may know, there are many steps. We've understood now about the genetics. We're starting to get clues about the disease mechanism, which giving us clues as to how we might look for drugs to support function when you don't have enough SCN2A, and also start to tell us about some of the considerations we might need in delivering this drug to people and running the trials we need to run so that we can show that there is indeed a benefit to these drugs, and that benefit doesn't come at great risk to the patients of doing more harm because these drugs have got their own issues that we need to deal with. And that's an important consideration in how you move an idea from a concept into an actual product that people can be administered to people and will deliver them some benefit. We're seeing more and more examples of this happening. We saw ASOs for Angelman syndrome, for spinal muscular atrophy. We're seeing molecules, small molecules, fenfluramine coming for Dravet and CBD. We're seeing other ASO programs from companies like Stoke that are looking at getting into that space for Dravet by making more of the SCN1A ion channels. So there's more and more activity happening in this space. Ultragenics is another player that's producing molecules in this way. So I think it's a time when many groups and organizations are recognizing that there's a real opportunity to do something now. We're doing what we can as a laboratory, and of my own involvement, which I have to declare in a company called Praxis. We're trying to do our bit to help patients as well. And I think, collectively, if all of these things come together, I think we know that there's great cause to be optimistic that something will happen. When exactly that'll happen is very difficult to predict. Who, of all the efforts, which one will have the most impact is also very difficult to predict. But I think it's great to know that we really got a deep understanding now of how SCN2A diseases emerge. We know about the clinical separation. We're starting to understand about the disease mechanism separation, and that thinking is starting to influence how we consider drug development. And now I think with that complete closing of all those loops, I think now it's just a matter of waiting and watching the programs develop over time and seeing which one comes to the fore, and hopefully we'll start to see some really great novel therapies emerge for these kids that we all care so much about. So thanks very much. I'll leave it there, and thank you very much for your attention.